This episode is brought to you by the Harry Butler Institute at Murdoch University.
Hey, I'm just quickly jumping in here before we get into today's conversation. If you're enjoying the podcast, please take a moment to like and subscribe. Click that little bell button to get notified when the new episodes come out. And if you'd like to support Paramount Importance even further, please check out our website, ParamountImportance.com. We've got some awesome merch there. They've got these cool shirts, these cool hats, all environmentally friendly and the highest quality we could go. It generally helps us keep on doing what we are doing, spreading the word of science, inspiring a whole bunch more people. It really helps us out. But without further ado, let's get into today's episode. Let's go.
Paramount Importance.
All right.
Yeah, well, after a few technical difficulties, we got there and we're going to get started. So thank you for joining today. I'm very excited to speak on this topic because I've done a fish parasite podcast with one of your colleagues, Storm. I spoke to Narelle about, parasites in feral animals. Mm-hmm. And today we're going to be talking about pathogens and parasites in zoonotic, which is one of my questions. What is a zoonotic parasite? But we're looking in humans, I guess.
Yeah, those zoonotic parasites are those that the term is they are transferred naturally between humans or animals and humans. So those things that can naturally move between animals and humans can cut across those barriers. And that's kind of where a lot of my research has been. So you just mentioned you talked to Storm about fish parasites. It's funny because in the parasitology world, we often, you go to a conference, you go, oh, that's the fish person, or that's a tapeworm person, or that's a protozoan person, where I kind of find myself as I follow the host and what's involved with the host or with the parasites of that host. So I'm a bit more of a generalist, I guess. But if I do have a focus, it would be within that zoonotic transmission, which takes me into parasites of wildlife, where I kind of really started all my work, and then into companion animals and livestock and humans. So it sort of allowed me to branch out into quite a few different ways.
Right. So when you're saying you're following the host, it's like because a parasite's life cycle can have many hosts.
Yeah.
So that's what you're saying. You're working like a very diverse area.
Yep.
And can take you everywhere.
Yeah. It does, and it has. I did another seminar this year. I can't remember now. But funny how I wrote my talk was basically that it seemed my linking with all of my research was about travel, and so I seemed to this involved me going here or this involved me going there. So my first ever research project, my honours year for parasitology, was actually at Singapore.
Right.
So I was I lived up in Singapore for a few months, and I was based at the zoo there, and I was looking at parasites of the giraffe that they have there, and they were having some health issues with a particular what they thought was a parasitic infection, and their giraffe were not very well, and some had died, and they wanted to know what it was, and so I spent 3 months up there collecting, and that's another commonality of my work is that I collect a lot of poo. So I was collecting giraffe poo daily at the night safari at the zoo, to the point where I can remember these giraffe, there was 3 of them, and there was they all had their personalities, and there was this quite dominant big male, and then there was a nice little female, and the dominant male really didn't like me. And I remember going up there after, many weeks of going up there and collecting poo. And I wasn't going into the enclosure. I was allowed. The zookeepers were doing that because they're dangerous animals. And so I'd go up there and I'm waiting. And I swear this time this very pompous male giraffe looked over at me, turned his back on me, pooed, and walked away. And basically said, there you go. Now leave.
Yeah. He's picked up the gist of the routine.
She goes quicker if I give her what she needs, and then she's gone.
Yeah, right. So what was the parasite? Was it a parasite that was killing him?
It actually was. It was, and this sort of led me into what I wanted. Well, further for the work, obviously, but it was an interesting case of a parasite that normally infects sheep. It's a known parasite. Farmers around here in West Australia would know. Its name is Homunculus contortus, or it's called the barber's poleworm.
Homunculus contortus.
That's the name.
Feels like a Harry Potter spell.
A lot of parasites are. anyway, it's a blood feeder, and it lives in the gastrointestinal tract of animals, or the abomasum, in this case, and can cause quite significant disease. So farmers have a real problem with them in sheep here in Australia, and it can definitely kill young animals. It's a natural parasite of sheep. It is not a natural parasite of giraffe. The differences in this scenario was the fact that, because another case is transmission of parasites does depend on, whether it's a natural parasite, but also the ability of that transmission to occur. So you need to know lots of different life cycles as to whether this could actually happen. Now, the life cycle of this particular parasite is basically a fecal oral, but the sheep pick it up by grazing in their paddock. Giraffe aren't grazers, they're browsers. In the wild, they won't be grazing from grass, and so they're not naturally going to come across this parasite. But in captivity, they don't have any natural predators, and so they were modifying their behaviour, and they were sitting down and grazing. And so they were picking up this particular sheep parasite that they weren't, as a host, used to, I guess, from an evolutionary point of view. They were a naive host, and so it was hurting them. And also another commonality with parasites in this scenario is that the treatment, we have drugs, anthelmintics, but there's a lot of misuse of drugs, and we get a lot of resistance, a bit like AMR, microbial resistance. But and so the drugs that the vets were using at the zoo were no longer effective, and so these parasites were just proliferating and causing disease in giraffe. But that led me to what would a giraffe normally get? This is in a captive scenario. What would they get in the wild? So I started looking at, parasites, literature around what was known in wild animals, and there really was not much. So that question, what happens? How do we find out more? And so that led me into a PhD, which I was never going to do, but I ended up doing a PhD, moving here to Perth, and running around the African jungles chasing the African painted dog, because that was a project that was there to look at between captive and wild animals. And so that's what I wanted to do.
Yeah, awesome. What a cool project too. It's strange though that you moved to Perth to then study in Africa.
I know.
Instead of moving to Africa to study. But I understand how that side of science works. What did you find with the painted dogs, and compare? So you're comparing captive versus wild populations of the painted dog.
Yeah, exactly. Yeah. So one of the reasons by moving to Perth was because we had the people that were working with these animals already. So here at Perth Zoo, John Lemon, who's the head. I can't remember now what because he keeps moving up the chain. But anyway, he had been running and organising conservation programs within Africa with a painted dog, and we also have the African painted dog here at Perth Zoo. So there was connections here at the zoo with captive populations, and there was connections with various conservation groups within 3 major countries I went to were Zimbabwe, Zambia and Namibia. And also South Africa, and also Adelaide Manato Zoo, because they had captive populations. So I had captive populations both in Australia and one in South Africa, and then I had wild populations in Zimbabwe, Zambia and Namibia. What did I find? What was expected, there was quite a difference between captive and wild. Captive populations had sort of, I did come across my first zoonotic parasite with regards to the painted dog, which was called Giardia, which is a protozoan parasite, which looks a little like this.
For the audio listeners, we have a stuffed toy. How would I describe that? Kind of looks like a upside down teardrop.
Yeah, the way I teach my students when I'm talking about this particular one is it looks like a little bit Casper the Ghost, because actually it should actually have 2 nuclei which look like little eyes. It has these things called flagella, which are these, tendrils hanging off them, and it floats around a little bit like Casper the Ghost.
Yeah, or it looks like a lot like a jellyfish.
Or a jellyfish, yeah.
And then is that flappy bit at the back also part, or is that to help it stick?
No, it's actually part of it, and also helps it stick.
Yeah.
But so this is called what we call a sucking disc on the back of this little protozoan single-celled organism. It lives in the gastrointestinal tract of just about every mammal I can think of, including people.
So we would have it.
Yep, and this sucking disc attaches to the intestinal wall. And that's where it can start to do some damage.
Oh, so it's not a good one.
It's not. Yeah, it's to be debated. There are many humans and animals that I know that are infected with this particular parasite that are asymptomatic, have no symptoms, they're not a problem, and others that are symptomatic, i.e. showing diarrhea. A common one that we as people would be familiar with this one, it would be termed as barley belly.
Right.
Yeah, so if you've got any diarrhea coming home from somewhere,
then
Giardia might be the criminal, might also be something else, but that's something that it can cause.
Right.
So I was finding there's lots of different species of this particular Giardia, and a couple of them are these zoonotic ones, so they can transfer between humans and animals. And so I was finding that in the African painted dog. In the captive population. So there's the question there remained, well, where were they getting it from? We think of zoonosis as, affecting us as humans, getting parasites from animals, but the reverse happens because it goes both ways. So was this a scenario where the dogs were actually somehow picked it up from, I don't know, someone, keepers or something like that sort of thing? So that question remains.
Right. What is your gut feeling on that question?
My gut feeling is that it potentially is transferred from humans. I think that happens a little. There's another really good story that I have with this one with the Giardia is in America. They often, so we will, call barley belly as a common symptom because we go to Bali a lot. In America, they call it beaver fever. And that's related to usually when people go camping in the national parks, they come home with, symptoms of diarrhea and they blame beavers for it. So they go camping and there's usually, by a water source and they've got lots of beavers around and they're blaming the beavers of carrying Giardia and then infecting them in the water source and people are in the water or drinking the water and they get infected that way. But in some cases, they have found that, yes, beavers did have this particular parasite, but it appeared that they were actually being infected by humans because they were downstream from a sewage outlet.
Oh.
So they were probably getting infected by people.
Is that what E. coli is? Because E. coli is like fecal matter in the waters.
Well, it's
right?
a bacteria.
It's a bacteria. Okay.
Yeah, but it's a similar kind of, so that's, so it would hang out in our gut as well. And so any things that are in the gastrointestinal tract will usually be found in poo, which is why I end up looking at a lot of poo. Because that's an easy way of finding the parasites. But yeah, E. coli is a bacterium which does the sort of same thing. And you've got some pathogens and some that are not. Yeah. So a pathogen I would say would be is something that causes clinical symptoms that harms the person or the animal, whereas a parasite doesn't necessarily always produce symptoms. So it's not always a pathogen.
Mm.
It's not designed to not be a pathogen, but in many cases, so a host and its parasite have been together for probably ever. Like ever since we had a host, we had a parasite. So there's been a very long evolutionary time where hosts and their normal parasites have been together. And they've been practicing this kind of little arms race going on constantly. Host, us, have our immune systems. That's our main artillery, I guess, against the parasite, and the parasite's evolving ways to outwit the immune system. So this kind of thing has been going on forever. And so when you've got sort of a natural host and parasite, it may not be as a significant pathogen because we've got that healthy balance happening. it will become a pathogen if able to, and that's usually if something happens to the immune system. And so if we're thinking about from a human point of view, so if we're something that's affecting our immune system, then the parasite that we may have will have more of an ability to be a pathogen.
Okay, I'm going to ask this question. What's the difference between a parasite and a pathogen? Like, how does a parasite become a pathogen?
If it starts, so I think of a pathogen as something that causes disease,
Okay.
causes symptoms, causes health implications. A parasite doesn't always cause disease. It depends on various scenarios.
Okay. When you were talking about, like, most of us probably have Giardia in our like intestines, is it like a genetics thing that some of us with certain genetics don't get
Symptoms?
symptoms from it, and then some do? Or is it just like the amount of Giardia that we have and then it sort of overrides our immune system? Or how does that sort of...
Yeah, look, it's an interesting question. And with regards to Giardia specifically, there's been a lot of research looking at why there is all this symptomatic versus asymptomatic cases. And there has been either there are more pathogenic strains or species of Giardia that are more pathogenic, so more likely to cause disease, and or it is different people's immune responses. So everyone's immune system is very different, and that can be a genetic background to our immune system, or it can just be our different makeup with, how stress levels are or how diet is or any of these things that may affect our immune system. And so those things mix together, and I don't think they've teased out exactly when and why Giardia causes symptoms in some people and not in others.
Right.
Did that answer the question?
Yeah,
kind of?
yeah. Well, it also opens up some questions
Yeah.
too. I'm going to jump back to the painted dogs.
Yep.
So these dogs that are in captivity, is that why they can never be released into the wild again, because they haven't built up the immunity from the wild population, parasites that they get?
So that was one of my questions in my thesis, and there certainly that was. I think that is the case in some cases. I found Giardia also in the wild dogs as well, wild populations. And again, that was me considering where these dogs are roaming through, and they do roam through communities and villages and things in Africa. So it was probably, again, picking it up from people. But in the wild population, there was quite a diverse range of parasites that the dogs had without obviously showing any symptoms. One of them was a hookworm, which we have lots of different hookworms. And there is a lot of research on hookworms that has been done, and they have seen that, particularly hookworms in dogs, at a young age, if they're exposed to hookworm, they can develop a level of immunity. At those animals that have not seen hookworm before and suddenly do get hookworm is another blood feeder. It causes quite severe disease in the GI tract, can die. And so that was then thought of that. So if we are having these conservation programs with the idea of reintroducing these animals out into the wild, then they potentially could fall over from the influx of all these natural parasites that they haven't seen before, and hookworm would have been one of them.
Right. Wow. Okay. I'm just wondering, as you said, reintroduction into animals, or breeding in captivity to do that, and that the success can be sometimes really low. This probably could have been a question I should have asked Storm, but you might be able to help. With fish, I'm assuming that, because restocking with fish in wild areas can sometimes be not very successful, and that's
potentially what's happened with the barramundi that we were just talking about.
Yeah,
because they've gone and put these fish on top of a place where there was the parasite, the intermediate host. But
so this is barramundi farming.
Yes.
Everyone gets the context.
Yeah, where these fish would ordinarily be able to move away from, and they've gone and plonked them on top of, an area where they can get reinfected. But go back to your initial question again. What was that? So it was about reintroduction or restocking and. So not necessarily, I haven't had much to do with fish, but we have had here at Murdoch University been involved with looking at parasites with that very question, translocating captive animals, endangered animals, not necessarily captive. So moving them from one place to the other. And the common one here in southwest WA is the woylie, a little hopping marsupial. And they were having big issues with their endangered status. So it had a crash, decline in population, and wondered whether it was disease and whether it was parasitic disease. And so there's a lot of investigation that went on into that. But besides that, they were also moving populations around. And finally it was coming into normal, not normal practice, but to be considered in practice, what are the parasites in the animals of where they originate from, and what are they got where they're going to go to, and is there a difference there, and is that going to have an impact, or do you want to have these animals take their parasites with them so that they're not going to be affected by anything new where they go? Because there's a couple of different things that go on there, but it isn't. There's more of a feeling there that the natural parasite fauna, particularly in wildlife, is natural. It's normal. A healthy ecosystem would have a healthy number of parasites, and so you want to maintain that normality when moving animals around. So either you don't want to take stuff from one place to the other, whereby the new population are naive, and vice versa, the new population coming in are getting exposed to other things. So they are looking at a lot more in that kind of field, and not considering that parasites are a pathogen, but are actually a normal part of the ecosystem, and they're a natural part of the ecosystem. With regards to the captive population, so like I said, I've worked with the zoo here in Perth, and now I still maintain a connection with them, and we run a diagnostic service, and I screen basically a lot of the animals in the zoo for parasitic infections, because their protocol now is not to eradicate parasites, but to just check the level and see if the level is getting high or low, and maintain it that way, because they recognise that they are a natural part of those animals and don't want to get rid of them all. But they also need to be able to be monitored to ensure they're actually not turning into a pathogen. So they don't actually get rid of stuff, which leads me into another world which I've done with Storm, which is like looking at parasites on endangered animals and looking at the co-extinction risk of the parasite going extinct with its endangered animal. They're the ones that actually are host specific to their little parasite, to the little host. The host dies, then the parasite dies. And we did this with the zoo recently with the western ground parrot, endangered bird here
very endangered bird,
southwest West. Yeah, so they were looking at a couple of birds in captivity, and they found some lice, some ectoparasite, little lice on their feathers, and they wanted to know whether these were a natural, like a host specific louse, like one that's just found on the parrot, or was it something that's been introduced from somewhere else, with the ultimate thought of, do we need to treat this animal, or do we need to kind of think about preserving the lice with its parrot? So 2 years later, we actually were able to determine, because taxonomy, which is Storm's area, we're able to determine that yes, it was its own little louse, and so we'll keep that little louse. And
how did you determine that?
Through both looking morphology, so the taxonomy, and also molecular work. That it hadn't. There's been some work out there, and we can see that it wasn't a generalist. It wasn't some generalist louse.
Yeah.
It was something very specific to this parrot.
Right.
So
Right. It's a strange thing to think about when you're conserving like the value of life for different things, right? Like it's easy to think. Maybe it's to some. I guess easy to think that like, yes, we got to conserve the parrot, but like, oh, why should we care about this little parasite? It's a species, but it's an interesting.
Yeah. And some of the pros for that, I mean, besides the fact you just, everything, they're an animal, they're an organism in their own right to be able to exist. But when I was talking about that sort of, host parasite relationship, it can be quite complicated. But those that have natural, had their long evolutionary time together. If we removed that host-specific louse, something else is going to jump on there and take that spot,
and that could be a pathogen. Oh, okay.
Exactly.
I'm seeing it now.
Yeah.
Yeah. Wow. Okay, that's interesting. Well, what are some of the more complex like life strategies you've seen observed with parasites? Do some of them travel like their life cycle is like a worldwide travel? I'm assuming ones with migratory birds or things like that, or
Yeah, life cycles of parasites. My students hate them.
why? Because it's complex. Yeah, okay.
Yeah, there's sort of three main strategies, I guess. But not all parasites are equal, and they all do something different. And without knowing the life cycle of the parasite, you can't do anything. You can't control it. You can't diagnose it. You can't treat it with any success anyway, because you need to understand how it gets from A to B and what you need to do to stop it getting from A to B, if that's your process. But yes, so you can have the direct ones that just, go from its host back to its host. And then you get those that involve an intermediate host. So they have a life cycle stage in another host. So when you're thinking of those blood flukes in the barramundi in fish, they will have a copepod and something else that's an intermediate host that will have to do a life cycle stage through. So that particular parasite in this scenario would need all of those hosts present for it to be able to continue its life cycle. Why that happens, don't ask me. It's one of these things that's evolved over time. So I guess one of the ones I've been looking at a lot for the last 10 years or so is more into the human parasitology world, is a parasite that transmits between pigs and people. It requires that, so the intermediate, or I wanted to call it the larval stage of the parasite, is found in the pig, and the adult stage is found in a person. It's actually a tapeworm, which is this
one here,
long thing that, for those of you who are listening, is a long, flat, white worm with an anterior end which has like little suckery structures, which it's a long piece of fettuccine is what we like to call tapeworms.
This has got stitching, and they're not segmented usually.
They actually, no, it does have stitching, and it's on purpose. It
is segmented.
Yes.
Oh.
Yes. So they have what we call, the segments are called proglottids, which are basically packets of, so a tapeworm is both a hermaphrodite, male and female organs, and so each segment is got the male and female reproductive organs, and they reproduce and they become these packets.
It's got lots of segments in there, so it's got lots of...
Yeah, they mature as they get older. So they start off little at the top, and they get bigger as they get down the bottom. And then when they get down, the bigger ones at the bottom are mature and ready to leave the nest.
Oh, they just like cut them off?
Well, they come out in human poo.
Right.
Yeah, and that's your now infective stage. It's full of eggs, and now that's ready to be.
They're just dropping like packets.
Yeah, little packets of eggs.
Oh, no way.
Yeah.
So it'd have lots of sexual organs, and then.
Yeah, so they're basically replications.
It's like a life cycle in itself on its whole body.
Yeah.
Wow. Oh, that is interesting. Scary. But interesting.
As a tapeworm itself is not really a pathogen.
They're the ones that can grow super long.
Yeah,
right?
that's why I have this really long.
Yeah.
Yep.
How long?
Oh, it depends on who they are, but I think I've seen some really good science programs. Michael Mosley was very good with some stuff, and he did a program on parasites. And if anybody's out there, you should look it up. It's horrific fun. And he infected himself. Oh, no, it wasn't that one. He did infect himself with a tapeworm, but there was another one where someone was infected with a tapeworm and they took some drugs so that they expelled their tapeworm and then they laid it down beside them. And I think it was about 4 meters long or something.
Oh, my God. Yeah.
Yeah, they can be. But they don't necessarily take up, so they're not as known as a pathogen, this little guy here, this long tapeworm. Because they just hang out in the gut and they absorb nutrients. They were, in the past, known as a diet pill.
Oh, really?
Yes. And so in the old days when they were travelling medicine man would be giving you a little concoction which had a little tapeworm cyst in it and selling this as a diet
No way.
pill. Yes, and it's still. So I was mentioning this. I looked it up at one point recently. So that's back in the old travelling medicine days. But it's still got some relevance today. I think people have done this before, and you can apparently buy yourself a little.
Still, that is crazy.
So because it doesn't do anything, it allows you to, take up extra nutrients so you don't put on weight.
When I was little, my granddad used to tell me, I used to have like 7 wheat bix for breakfast.
Oh, yeah.
You
You had worms?
Yeah. And he used to always say, "Oh, you're an expensive one to keep." And then he was like, "You've got worms, boy."
Yeah.
Is that referring to a tape?
I think so.
Okay. He was a farmer.
Yep.
Yeah. So
I remember the same thing as a kid as well, being told that. But or you get worms from that, which now I know. I go, no, I wouldn't. Don't get it that way. Now I know. But
so you say you get it from pork.
So sorry, back to my story. I'm getting way late. So the one I'm working on is called a pork tapeworm because we get it from eating pork or undercooked or raw because it needs to be still alive to eat. And so that's the general life cycle of many tapeworms. It's the animal or the person that has the tapeworm has to eat something that's got the larval stage, and that's the way it goes. Does that make sense?
Yes. Yeah.
And that's normally fine. This particular one, however, the pork tapeworm, which its name is Taenia solium, is people can also infect themselves with the larval stage. So not just a pig with the larval stage. So and that tends those cysts, those larval stages, which go and cyst in the musculature somewhere in a pig. In humans, they tend to go to the brain and cause epilepsy. And so in countries where Taenia solium is endemic, not Australia, it's thought that up to 30% of epilepsy cases are related to that particular parasite.
Whoa.
So that's one I'm working on I have been working on for the last 10 years or so in Laos, in Asia, because it's a country that has all of the transmission stages that can occur, and they have the parasite, and so it does occur, and it's very hard to control.
Yeah. Is it just Laos? Is it other countries as well?
It's endemic in a number of other countries. Other countries within Southeast Asia, Central America, South America, and Africa, there are countries that have this particular.
How common is it for like travellers to get something like that?
Well, you'd have to. No, it's not common, but it can occur. Another one which I usually throw up at the students is this. There was a case where it was an Australian rock band, Frenzal Rom. It was a while ago. This band, anyway, punk band. They were traveling around America, I think, or Central America. Anyway, so the lead singer was put in hospital because he had a tapeworm in his brain, and he was saying in this article that, well, I'm a vegetarian. How did I get this parasite? Which is a common misconception because, yes, you get the adult tapeworm by eating meat, but you get the larval stage from these little eggs
that are coming out.
The
bum.
Exactly. So the proposition there was probably the chef who prepared his salad out the back may have contaminated his salad. Hence how we got infected.
Yeah, okay. There were possibly other ways too, maybe, but yeah.
Just when you're saying, but I'm a vegetarian, I don't eat meat, it doesn't matter. It was whoever. So shaking hands or, just that sort of thing. So
right, wow.
Yeah.
Okay.
Yeah, so it can occur, in that way.
How is it curable? Well, obviously he didn't die, I'm assuming.
No, he didn't.
You don't just live with epilepsy for the rest of your life. You can
and I don't actually know whether all cases do lead to epilepsy anyway. But it's just the fact that these cysts end up going to the brain. Yes, you can take. There's medications for it, are quite good medications. So it is. If I have heard of and seen cases where there are just far too many parasites in the brain, for instance, that the person has not been able to recover. So it has and can cause death, but it is certainly treatable.
Mm. Where has your research taken you across the world? Like, I'm assuming many places.
Well, the main ones are Africa and Asia. So yeah, like I said, with my PhD, I was running around chasing the African painted dog across a few countries. And this one's kept me in Laos for quite a long time, the tapeworm one. So that's sort of the main areas, and then of course in the Singapore Zoo as well. Otherwise, I run around Australia as well, chasing things in dogs.
What has been some of the more deadly pathogens that you've worked with, and has it, like, when you've been in these environments, has you been worried about your own health, or has there been like?
Yeah, the more I've learned about all of this world, I think before I wasn't always a parasitologist, and in my younger days I travelled a lot anyway, and now I look back at my travelling days and think, how did I manage to get through all of that uninfected? Now that what I know, I am aware. So when I'm travelling, I make sure my food is cooked. I do not eat anything that is undercooked. I'm a little bit wary now of lettuce and what I call liver fluke lettuce. It is what it is. In these places where I have been, when I feel like I'm at extreme risk, so I'm actually, dealing with biological samples that are potentially infectious, and we always use the appropriate, PPE, even when I'm in the field.
Like the full hazard suit?
No, but I'm in really remote areas, so I'm literally, sitting outside somewhere. But I'm always got my gloves and my masks and all that type of stuff. And I come home and I ask my GP for some dewormers.
Right. Okay.
As a prophylactic kind of treatment.
Right. And you haven't been in like too worried when you've been in these areas about, it's like you've taken all these precautionary steps, although, like, has there been some times in like places that's been like do you get sent to places when there's been mortalities, and like what's causing this?
No, I haven't. But when you think of the danger aspect, I think the most dangerous aspect was probably during my PhD, because I was there's a whole lot of other animals where I was. I had hyena taking my samples. I've had lions walking through camp. Various other things elephants sleeping next door to my tent. Hippos, having to have a hippo roadblock between me and the toilet in the middle of the night. But yeah, so those were probably more dangerous aspects, I think, rather than the parasites themselves. The parasites are friendly. It was having to navigate.
Cool experiences, like a person that's sort of, I guess, going into university, studying, and wants to sort of like use their degree to travel the world, parasitology. You wouldn't think that
could be.
First thought would be like that's going to help me travel the world and go see all these cool animals and all that, which is it's a really unique thing. Which environmental reservoirs or intermediate hosts pose the most difficult challenges for parasite control programs?
That's a good question. Depends on the scenario. I mean, like for instance, this little tapeworm that I was talking about, the Taenia soli and the pork tapeworm, we have a toolbox of control. We know what the drivers are. And we know the life cycle, so we know how we could potentially stop it. But the environmental factors and the context of where this is happening is difficult to control. So, for instance, in the country of Laos, when we're dealing with this, we know that communities don't have a lot of very good sanitation, so hardly have any toilets. So, therefore, that infective stage can get into the environment. They have free-ranging pig systems, smallholder farming systems where pigs are roaming through villages and cleaning up everything, so they're exposed to the infective agent. They then have deeply entrenched cultural and traditions that involve eating raw pork or a very undercooked version of pork. So these transmission cycles, it works perfectly But if you go in and treat a lot of people, you've done nothing to the pigs. If you go in and treat a lot of pigs, you've done nothing to the people. Transmission cycle keeps going. This is what we call One Health. I don't know if you've heard of this term before, but more and more being pushed that we need for these types of zoonotic parasites, you need a One Health approach. You need medics, you need vets, you need an environmental aspect that goes with it as well. And so in this project, I'm a parasitologist and I had to have social scientists involved because it's about behaviour change and how would you implement behaviour change around people who are not seeing the problem. Why do you tell me I can't have my traditional raw pork that I've always had? I don't see the reasons why I need to change, sort of thing. So that's what I mean by which is the most difficult to control. It depends on the environment, and I feel like those that are entrenched into traditional behaviours would be the hardest. But from a reservoir or another point of view, another common one that everyone would be familiar with would have to be malaria. So that's a protozoan parasite called Plasmodium that uses a mosquito as its main host.
Isn't it the most deadly thing in the world?
It's the most deadly thing in the world. And there's been millions and billions of dollars spent on trying to find control mechanisms for malaria, none of which have worked because it's far too hard to control mosquitoes. And they've only just come up with a vaccine, and I think they've spent about 60 years trying to find a vaccine.
Oh, so there's one.
There is one that WHO have just
How recent?
2 to 3 years.
Okay.
That they are promoting that can be used in countries that are required. But all vaccines have their issues, and how many doses do you need to have, and what ages do people have to have them by, and how they're going to get them out to the communities that need them. And I think it only has about a 50% efficacy. So it's not super great, but it's the best they've been able to come up with so far, because parasites are tricky.
Yeah.
They are tricky.
Adaptable.
Yeah, exactly. So
there's like malaria also in WA. I was reading one of your papers.
So that's not my work. That's my PhD student's work,
Yeah.
Ashley Peck. And she's just published a paper. She's been working on looking at blood parasites in mosquitoes around the urban regions of Perth. And she was finding a lot of avian plasmodium, so avian malaria. It's not going to infect people. Because there are other malarias or plasmodium species that infect birds, reptiles, they're the main other hosts. Otherwise, we've got human malaria. So it's not going to infect people, but it's an interesting find. We know hardly anything. When we get to wildlife parasites, we really don't know much at all. We don't know necessarily their life cycles. We don't know if there's another intermediate host involved. We don't know anything. So she's finding the avian plasmodiums in all these mosquito species, but we don't know which birds they're using as their hosts because we haven't matched that data up because no one's done it. So
Right. Oh, interesting.
Yeah.
How does because at some stage, right, a pathogen or the parasite can become a human pathogen?
not always. This is where we.
But say for was coronavirus a pathogen? Well,
COVID.
Yeah.
Yeah, it was. So virus, it was thought to be a zoonotic pathogen or virus. So therefore the transmission they think came from animals into people.
Right. But because it's new, right? Is it not? It was new. So how at some stage does this or even a different one now. I know that the avian influenza, the one that's killing all the birds around the world, and some mammals. I know a lot of seals and things have died as well.
Yep.
We're a mammal. They're saying
Yeah.
we're not worried about it yet, but it could. How does a parasite then change to be able to adapt and then start?
They're not as quick adapters as viruses are. Viruses have got generation turnover like crazy, so they can then evolve a switch a whole lot quicker than parasites can. And look, there have been the odd what we call a host switch, whereby some sort of pressures have pushed the parasite to do a switch. Doesn't happen often. So yeah, they're not nearly as problematic when you think, oh, is this parasite suddenly going to become a human pathogen? Because they're fairly entrenched in their life cycles, and it's a lot trickier for them to adapt out of that life cycle.
Oh, that's good to know.
Less of a concern.
Yeah. Is there any positive impacts to humans, like having a parasite or into the environment?
This is almost my favourite topic.
Right. Okay.
This is something got me involved with interest in parasitology. Full stop, really. So remember I talked about there are definitely some potential positives of parasites. So I talked about that really long evolutionary interaction with their host. And as humans, we have had a very long interaction with a lot of parasites. Now, those of us that live in lovely, clean, developed countries have no longer really got any of the major parasites that are commonly seen in other developing countries. And so if we consider from an evolutionary point of view, we've got lovely clean toilets and we took some drugs and we've maintained that for the last 60 years or so probably. And there seems to be a direct correlation with the lack of parasites or pathogens and the increase of autoimmune disease.
Right.
So the theory is that we're too clean, the hygiene hypothesis, too clean for our own good. And at a young age, particularly if our immune system is not developed and taught what is a pathogen, what's not, who to fight against basically, and who not to fight against, it goes out of whack and crazy. And so that's what all our autoimmune diseases essentially are, is our immune system overreacting to something it shouldn't react to because it doesn't know what it's doing anymore.
Is it also in like allergies as well?
Mm-hmm.
Yeah.
Yep. So one of the things that they did show was, and there's some really nice research that is continuing this up in JCU in Cairns, but there was, have you heard of the disease Crohn's disease or ulcerative colitis?
I've heard of Crohn's, but I actually don't know what it is.
So it's an autoimmune disease, and it's basically the immune system attacks your own gastrointestinal system. And so people who have Crohn's can't, have very sensitive GI tracts. They can't eat anything that's of any spice. I've had a number of friends in my environment who they're basically living on boiled rice because they can't eat anything. They have multiple surgeries often because their intestinal system is so ulcerated it needs to be taken pieces out. And this is just immune system that's going crazy at itself. And so going along with this hygiene hypothesis, some scientists in the States back in the 90s trialed it, and they put a parasite, a very generic parasite, it was actually of pigs, but into people with Crohn's disease, and they were symptom-free whilst they had their parasite. The theory is there that, we've had immune systems, had a parasite to play against forever, and we took them all away 60 years ago. That's a very short period of time in an evolutionary point of view. So the immune system's going, well, what am I supposed to do now? You've taken all my playmates away. Yeah. I've got nothing to fight and I don't know what to do. So it attacks itself.
Right.
So by putting parasites back in.
So is that when they do the fecal transplants?
Little different. But I think is
that more microbial?
It's more about, getting a healthy microbial kind of content so that everything else is working fine. Okay. But this is actually because the main thing that parasites do, or the main instrument that they attack, is the immune system. That's our arsenal. So they're also potentially genetic drivers as well, because you've had this co-evolutionary arms race continually going along, and us as a host having to develop something else that outwits the parasite, and so it continues, and that's our immune system. So our immune system has been maintained, a healthy immune system, we could say, has been maintained by a healthy burden of parasites. Take them away, we've now got lots of autoimmune disease.
Right. What are some other the autoimmune diseases that people would know?
So I talked about this JCU crowd that are working on it, and they're looking at celiac disease, have looked at asthma, they have looked at I think multiple sclerosis as well, that they are looking at they're calling it helminth therapy, I think now.
So they're all related to not having parasites,
Yes.
potentially.
Yes. I think it's multifactorial. I think parasites are part of it, but it's basically taking away any pathogen for the immune system to work out what to do.
Wow.
Yeah. So there's
That's
the
incredible.
good side of parasites.
Yeah, I've never heard that one.
Huh.
Well, and what about like, I don't know, I'm going to use myself as an example here. I grew up in the bush, very grotty, rubby, running around the farm where I've moved to Perth. Perth is obviously a lot cleaner than where I grew up. Can you develop in your more adult ages of life like these
autoimmune responses?
Like if does my immune system can lose the ability to fight these different but they're not pathogens, right? They're in there just
back to the pathogen versus parasite.
Yeah.
For it to become
Like can I all of a sudden become celiac?
Yeah.
If I like spend 20 years up here. Well, I've only been up here for I think 5 years or 6 years, but
I don't know the answer to that question. I suspect though that because I had a probably similar upbringing as you, that put us in good stead to have a nice, well-developed immune system that's going to happily do its thing as we go along. On saying that, people do, develop celiacs, later in life. So I can say this, I think. So I was talking about this group at JCU who are working on using hookworm, I think, for instance, as a helminth therapy. And there's this great researcher, and I was talking to him at recent conferences, and at one point I was talking to him about his work, and he said, and he had his PhD students around, and they went, oh yeah, we've currently got hookworm infections, because they were doing part of the trials that he was working on. I don't know how he got that through ethics, but anyway, he did. And at a recent conference, I was chatting to him, and I said, how's it all going again? And he has his own hookworm infection, and he's using it, he thinks, to combat arthritis. So he said he started getting arthritis in his hands, and he wondered what should spruik what he's talking about. So he said he's been maintaining a hookworm infection, and whilst that's happening, he doesn't have as bad arthritis in his hands. So
is this causation or correlation?
I know, right? He's the researcher. I don't think there's enough empirical evidence there, but they're working on it. They're really working on what does a parasite stimulate.
Yeah.
And then if they can work out a synthetic version rather than having to infect people with parasites, for instance, but understanding what is it stimulating, how is it stimulating the immune system, and can we somehow develop something that can then help these other people who have got all these autoimmune diseases.
I know this is not quite in your research, but like, do you think that for a lot of like cures and of the future, like is going to be parasitology work or parasitology work? Yeah, parasites curing things, because like all those things there sounds like the reasons we are getting these autoimmune diseases or potential or parts of it, and then like if this guy, he would probably win a Nobel Prize if he can cure arthritis by infecting yourself with hookworm. Like there must be obviously more work putting in these to test these things. It's a strange way to work this out, I guess.
And I mean the difficult part I think that they're finding is that because the host parasite interaction is so complicated. That's been one of the biggest issues about trying to work out how a vaccine might work, because there's just so many different stimulations or antigen things that are popping up during different life cycle stages, all this sort of thing. So they're very tricky. So I think getting something to standard to replicate is where they're struggling with, but they're identifying a lot of responses, I guess, host responses that they can potentially make as a therapeutic of some sort. How far parasites go in that field, I don't know. It'll depend on how replicable it can be.
Yeah, it's super exciting. Like to know that there's all this crazy work, I guess, still happening, and like things that we can find out. And like, I remember I did a podcast with Navid about algae, and like the the applications that can happen in the future from that, like it's all new research and like, wow, that can solve so many different problems. This sounds like it can solve so many different problems as well, which is like, wow, the world can change. Obviously, we're probably not going to solve global warming from either of these, but like super, super interesting.
Yeah.
Like,
and they're usually not direct outcomes from a particular research project, so it's or in many cases we get these scientific revelations out of something that we didn't know we were looking for.
Like penicillin and things.
Yeah,
right?
exactly. And now, I mean, the funding environment is just so difficult, and you can't get funding unless you are going to try and combat global climate change or something, without these kind of kooky little things happening on the side. So the funding scenario is just getting harder and harder. So.
It's getting scarily worse, and that's global politics, really, right? I guess.
Yeah.
I'm going to jump back into some of these other questions.
Okay.
Because I realised we've only asked probably about 4 questions.
Sorry.
No. It's been so interesting. Like, hence why these are just act as a guide, and we just go wherever the conversation goes. So we've talked a bit about Giardia. Did we talk about the Cryptosporidium?
Cryptosporidium and Giardia. This is my little Giardia man. Cryptosporidium and Giardia, no, we haven't. They usually go hand in hand, those two. They are they're both little protozoan parasites. They're both transmitted the same way. We usually get infected by contaminated food or water, and they give the similar symptoms. And they're global. They're everywhere. And they are probably one of the last few parasites that we would have across the board here in Australia and around
Parasites
the
or
world.
pathogens?
Well, they are both. They're both parasites, but when they become a pathogen.
Yeah. Okay.
So I'm not an expert in Cryptosporidium. We've had researchers here. I don't know if you've spoken to Professor Una Ryan, who just recently retired, but that was her research world was Cryptosporidium. I do have a student who's working on it that was with Una as well. It's, like I said, it's often a water sort of contamination scenario. It's still a fecal oral transmission, so it's just contaminating water or food with that. And pools are often the big risk factor.
Really?
Yes.
So the chlorine in pools doesn't quite kill it?
Yes, that's correct.
I spend a lot of time in a pool. I'm doing an Ironman in a couple of weeks.
Oh, really?
And the amount of training in the pool is a lot. I'm always like, some of the weekends I go there and there's like, they set up these bouncy castle things for the kids, and I'm just going like, oh, a lot of kids in the water. There's probably a lot of things I'm like, oh, it sucks. It's in your mind. Don't swallow anything.
Yep. I mean, Cryptosporidium is one of the notifiable diseases in Australia. I think it has to be correct. I could be wrong. But the outbreaks that do occur, it must be, because we get, Una has been chasing these types of things, outbreaks that are usually associated around pools. We have just chased a recent outbreak in Perth earlier this year. And our PhD student was genotyping that to understand. So this is where sort of where you have to do this type of work to understand transmission pathways, where there's different subtypes and things, and they're trying to find out where it's coming from. And we've just got that recently published, but it's a new subtype that occurred earlier in the year. We don't know where it came from, but it was definitely correlated with pools and childcare centres. So put those two things together. And you've got a nice little place of Cryptosporidium. So, those bouncy castles that you're swimming around
right.
with what's called the accidental fecal release into the pool.
Right, thank you. That is going to be in my mind.
Sorry.
No, that's okay. Well, you've touched base, like, why it's important to study the life cycles and where it comes from to be able to, obviously that's the main action. Is that the same, like, I guess the question here is, why does understanding the genetic diversity of pathogens, is that the same as the reasoning for studying the life cycles, like of those two parasites you just mentioned, and Yeah.
Yeah. So genetic diversity is sort of twofold. It can either be chasing, an outbreak of disease, so like Cryptosporidium outbreak, to understand where that has come from. There was a good classic story we talk about with Cryptosporidium, which came from the States, where there was a massive waterborne outbreak in Milwaukee. A quarter of the population were infected, like 400,000 people. And there were deaths involved as well, particularly immunocompromised people. Cryptosporidium has been quite deadly. And they were blaming cows for contaminating the water from effluent of cows, because the cows can carry a Cryptosporidium. By genotyping and going in and looking to see the genetic diversity of what was the Cryptosporidium that was being spread at that time, they actually traced it back to the fact that it was human effluent that was in getting into the city's water source. So it wasn't the cows; it was humans. So So that's probably the important aspect of genetic diversity. I guess the less important from a public health point of view is just understanding, the number of different species of parasites that are out there.
I guess when you talked about the ground parrot before, even though they said it was morphology and taxonomy that determined it, right?
It was a bit of both.
Of both.
Yeah. We need both now. We actually, molecular work is becoming more and more important with parasitology. But without the taxonomy, though, it's almost useless. So one of the things I had when I was doing the African painted dog work, I came across some tapeworms of the African painted dog in the wild, and I didn't have enough information to do taxonomy on them. So I was genotyping what I tried to see what the species was. So I got this lovely sequence read, and you go and compare that to published sequences in our genetic databases, and it couldn't compare to anything because it's never been done before. So, if you haven't already described the organism and got the genetic data to match it, put it up into these public databases so the next person can come along and say, oh, yeah, mine matches yours. You described it. It was this. But I've put my new stuff up there, which no one has got. So it doesn't tell me anything. It just tells me I've got something different. So that's where we need a lot more information when it comes from a genetic point of view, whereby we now try and.
Did you describe that species?
No, I still don't know what it was. But we did it. We have done a tick as well here in Australia. We were one of the first tick in about 50 years, a new tick that was described from our lovely little friend, the woylie.
Right.
And that required the taxonomy and then the molecular as well.
I heard this thing, I hope this is true, that ticks use static electricity to jump onto other.
I don't know.
Yeah, well, I'm actually pretty certain it is true.
Yeah.
I think it was Joe Fontaine who told me this actually. I don't know if all ticks do it or for a specific one, because they can't jump. They don't have the hind legs to jump onto animals or things like that, but they can use static electricity to, like, I guess, in a way, jump.
Yeah, exactly.
I don't know how else to explain it.
I don't know. I've never sort of thought about that one. That's pretty cool. I'll have to look it up.
I'm pretty certain. I'm almost 100% certain it is true, because I think I did even see it on a BBC thing.
Did you?
Yeah, which was like, wow,
fabulous.
But I don't know how many ticks, if it's all ticks or not.
Yeah.
Very interesting.
Yeah. There's so much we don't know.
Oh, it is. Especially, parasites is one of the largest groups as well of, like,
yes,
in the animal kingdom.
Yes. Yeah. Because pretty much every host has got a number of parasites. So, and it's not just animal hosts, you can plant hosts and all sorts of things. So, yeah.
If you were to say to a student, why should they study parasitology, what would you say?
Because they're cool.
Because they're cool.
Why? Because the students either go one way or the other. They either really love them or they go, they're too icky for us to worry about. But I think it's because there's so much going on. Well, I like to call them infectious diseases with personality.
Okay.
Because They've got personality. They've got a different life cycle, or depending on who they are and how they get there. And it's also a little bit of a detective story if you're going in there as a control, which is a lot of my work as well, is thinking about how to control transmission. And you have to know all of these things to find out how to do that. So it's not just, it's this holistic look at everything to try and so you get to learn a lot of things, I guess.
Yeah. Well, I got to admit, when I first started learning about parasites, I was a bit like, oh, that's a bit like gross. But like the more I've learned from Storm, from Narelle, like Storm gets to go travel all around Australia spearfishing. That's my dream.
I know, right?
How cool. You've traveled the world. Narelle does all as well, travels all around in the bush, getting feral pigs, foxes, cats, all that kind of like. So you get like the element of travel, doing some cool field work. You've got, and then also you potentially can win a Nobel Prize for curing human diseases, or like
there's that.
The fact that you can save, I guess, a huge population. So there's like
there is a lot
very important.
Yeah, we like to think so. Thanks.
Yeah, well, I didn't think how like I remember when Storm was coming into the lab collecting parasites, and I was just like, it's strange. What are you doing? What application is this going to have? And then learning about it, it's like, wow, there's actually there's so much. I'm like, now I'm thinking like, oh, no, what application does my work have?
Well, you can put parasites in there if you like. I mean, it's just so many stuff. So we sort of do a lot of feelers with lots of different people, but we've got an intern in pathology, vet in pathology at the moment, who's looking at western ringtail possums and finding parasites there, and so he's bringing them up to us to have a look at. And we're like, well, okay, that's great. Yeah, we know something about this, but we know nothing about the other part of it, and we don't know how. And this is critically endangered animals. That is a parasite a pathogen, or is it just a natural part of its system? So so many questions, so many things to do, and you can go and if you want. I guess I try and say to people, are you can you travel with that? I think if you want to travel, you'll find ways to fit your research into that anyway. So it's not like necessarily a direct, thing to do, but I like and can, and I stopped telling my supervisor during my PhD what sort of risks I was up for in the field, because it was just crazy. But also, if you're not that inclined, there's a lot of really cool laboratory-based stuff that can be done as well. So, designing vaccines or, new drugs, therapies against Cryptosporidium, for instance. We don't have any useful medication. No one's been able to find one yet. So there's a whole another area. Or you can go looking at parasites as a therapy. It's got lots of different angles. Yeah.
It's been absolutely super fascinating. Thank you so much for coming to join me today.
Thank you, Kurt. Thanks for inviting me.
Yeah, I was, after when I met you after the Sustainable event, I was like, you're coming on.
This production was recorded at the podcast studios in the Media Arts Centre at Murdoch University. The views and opinions expressed in this podcast are solely those of the hosts and guests and do not necessarily reflect the official policy or position of Murdoch University. Any content provided by the hosts and guests are their personal opinions and are not intended to represent the views or opinions of the university, its administration, faculty, or staff.